we are nearing the end of a long course on systems biology, and these are some last topics that remain in the course. So, today we will look at a very interesting paper that talks about bridging the gaps in systems biology, and basically what are all the challenges in systems biology, be it model standardization or model composition and so on, and how the need of the hour is more interdisciplinary research and students who are comfortable with both quantitative and biological subjects, right. And I will also talk to you about Dream Challenges, which is a very interesting consortium that proposes challenging biological problems for the community to tackle, and we will have a brief overview of wholesale modeling, and finally I will talk to you about an interesting concept known as retroactivity, which has very important implications for circuit design in synthetic biology. So, welcome back. Let us begin concluding the course, wherein we will try to summarize a lot of concepts that we have looked at so far and touch upon some important open challenges and some interesting research problems that are going to occupy the field for the next few years. So, I will first start with some of the interesting open challenges and so on. There is a very nice paper that I have shared on the class, which is called Bridging the Gaps in Systems Biology. It is authored by literally the who's who of systems biology, and with a lot of engineers, biologists and so on, right. And I am just going to emphasize on few of the interesting problems that they have talked about. The first of them being something that we discussed rather recently, which is how do you handle uncertainty in systems biology, right. So, they say that when building mathematical models of biological networks, at least 3 interlinked levels of uncertainty can be encountered. Uncertainty in network structure, the topological uncertainty as we saw previously, the uncertainty with respect to choice of kinetics, right. Should you use mass action, should you use Michaelis-Menten, should you use Hill and so on, and of course uncertainty in the parameter values, right. So, broadly speaking you can talk about the first and the last, and the choice of kinetics is somewhat intermediate, right. It mixes some aspects of topology and some aspects of parameter, right. So, this becomes quite a big challenge. How do you pick the right model? And this is something that people grapple with all along, right. If you look at we will also have a few right, but if you look at the field of quantitative systems pharmacology, you will find that they have this problem day in and day out. I have some data coming in from some literature or some carefully constructed experiments. I have a model. When do I know that my model is fit for purpose or not? Can I if I am not able to fit my model to this data, does it mean that A, my parameter estimator is no good, or B, my model itself is of the wrong structure or so on, right. This becomes a very difficult question to answer, and we want to fundamentally answer these kinds of questions. It is quite important. And then one very other very important concept in systems biology is that of model reduction or model simplification, right. So, how do you simplify the models? And this becomes quite a challenge. So, model reduction or model expansion. So, how do you simplify models? So, and as they note here, drafting a model from mechanistic hypothesis often leads to models that are very large with large number of parameters, right. This is a classic issue that we did touch upon during dynamic modeling. You essentially know that there is this particular reaction involved, there are these species involved. So, immediately write mass action or Michaelis-Menten for all of these. The moment I write out a Michaelis-Menten equation, I have 2 parameters per reaction. So, my system just explodes. If something is really fast, you may just ignore it, right. That reaction is as good as you can just ignore that reaction from the rest of the mix, right. So, these kinds of approximations have to be done. Can you do them systematically? How do you systematically simplify models? So, this becomes very interesting, right, and very challenging. How do you bridge different scales of modeling abstraction? But as we did see, some discrete modeling framework can come in very handy there. Merging models, this becomes very difficult, right. So, if you have multiple models of the same system or of related systems, can you wire them all together and try to simulate a much larger system? This becomes quite challenging, beginning with the fact that the nomenclature may vary between different models and so on. This is something we encounter a lot when we look at community modeling using flux balance analysis. So, when you try to combine genome scale metabolic models that have already been well curated, well studied, even validated, you will still find that you can't easily marry the models because there is no compatibility in terms of the annotations that are used, the annotations may be missing and so on and so forth. This is something I briefly pointed out to you when we were looking at flux balance analysis as well. And of course, this becomes a challenge too. Each individual model is designed for a particular purpose using some specific data, and maybe it could be at a different level of abstraction. So, how do you merge them, right? So, in a big challenge in multi-scale modeling comes from integrating across time scales, integrating across spatial scales and so on, right. How do you integrate across these, and the frameworks may also be different. This is a huge challenge. We really need good standards in the community and standards that need to be adhered to. So, SBML makes a set of recommendations, SBGN for drawing, making diagrams makes a set of recommendations, but you will see that they are not as widespread, followed in as widespread a fashion as they should be perhaps. So, they say that even when appropriate experimental data exists, it can be difficult to use them due to limited or inaccurate documentation and annotation. So, you need standards for everything. If you have microarray data or RNA-seq data, metadata is so important, right. If you need to expressly state what kind of conditions you created this data under, what were your exact experimental conditions, and these are usually not communicated from experimentalist to modeler, and this becomes a challenge. And invariably you will see that when in fact those of you who are doing your projects, you will find that you are not able to reproduce experimental results, because there is always some data that is missing, right, or some concept that is not been enunciated very clearly in the manuscripts. And this is of course my favorite challenge. How do you bridge the gap between wet and dry, right? Do you teach biologists more computation? Do you teach computational people a little more experiments and so on? I think you need a mix of both. I think as a modeler you should be aware of what kind of data is coming in, what is the data that you are working with. You should have a very good understanding of that. Without that, I do not think you can build good models. This is why invariably the one important wisdom in the field is that the same person should somewhat try and do the modeling and the experiment, because then you will know, I think there was something wrong with the instrument the day I was doing the experiment, right. This could be the reason for this outlier, right. So, these kind of insights are impossible to get when somebody gives you an Excel file with all the data in it, right. But if you have, when you have done experiments, you will recall that there were some issues, or there are some possible sources of variation. So, this is going to be a challenge, but as long as they are documented well enough, it could be a middle ground. So, then the experiment may also reveal a large number of interaction partners, but you do not know the context, right. So, you may have an experiment which tells you A, B, C, D, E, F interact, but maybe they observed it under certain specific conditions which does not apply to your scenario, so on and so forth, and so all these become a little challenging. I really like this as an important message from this course for all the people who are doing this course and even doing it online, is in particular graduate students will benefit from learning by doing interdisciplinary research projects with dual supervision with mentors from complementary disciplines. So, I think this is very important. So, those of you who venture into research or venture into the field of systems biology, I think it is nice if you can find two advisors, maybe one with more of a computational background and one with more of an experimental background, to help you guide through projects that are interdisciplinary in nature. And they say that the future success of systems biology largely depends on bridging the current gaps between different scales of modeling abstraction, topological and mechanistic modeling, and wet and dry labs. Right. And the development of standards of both modeling and experimentation is crucial for facilitating model reuse and for increasing the pace of mapping out, describing and better understanding biological systems using computational approaches. And this basically transcends the kind of modeling approaches that we use, the kind of programming languages that we use, the kind of software tools that we use, and we used quite a bit of all of these in this course, right. We used a bunch of modeling approaches, a bunch of programming languages and a bunch of software tools, right. So, how do you reason out, or how do you reconcile between all of those? And I think you need to have a good understanding of all of these techniques to essentially make good inroads into systems biology research. So, So this is roughly, the epilogue of the course, wherein we are just trying to wind up and say what are all the important concepts. I will try to do a recap of some of the most important concepts and so on, but this is roughly what I wanted to cover. So and I am also happy to answer any questions related to systems biology, right. So things we covered in the course, things we did not cover in the course, right, or if you say I have heard about this modeling technique, where could I learn more about it, kind of thing, right. So I will just keep the class open for a few minutes for that, and then I will maybe start trying to do a recap of the entire course, a quick recap of the entire course. In terms of practical applications of systems biology, they are in many different fields. So the first application is in biology itself, right, trying to better understand biology. I think that's one major application of most of the modeling approaches that we saw and so on. And of course, clearly in pharmaceutical industry, and metabolic engineering, so on and so forth. So either drug target identification or improving the strain improvement for metabolic engineering and so on and so forth. I think those are examples that we very well studied in different modules of the course. And in general, things like aspects like network biology are useful across the table. So there are insights from systems biology that are used in, say, social network analysis and vice versa and so on. So I think there are other such very interesting things. One more thing that we could probably look at is, I think this is something that you should all take a look at, the DREAM challenges. So DREAM actually stands for some dialogue for reverse engineering and applications in modeling and so on. Some acronym made out of that, but essentially they have a lot of interesting challenges that are involve systems biology, machine learning, computational aspects of whatever we have discussed. So for example, they had a parameter estimation challenge some time ago, they had a disease module identification challenge. So I think this is only the open challenges. So if we can look at closed challenges, you will be able to see a lot more interesting challenges. So these are all very interesting projects that one can do to, you can get your hands wet with in systems biology. Right. So in vivo transcription factor binding site prediction, digital mammography, this was more of image processing and so on. Let's maybe the disease module identification challenge. This is a challenge in which we actually participated and performed reasonably well. And drug combination prediction challenge, it was truly challenging to solve that. Very interesting challenge on olfaction prediction. So they had chemical molecules and you had to predict the smell of a new chemical molecule by learning on previous chemical molecules. And some more classic ones like parameter estimation. This is a gene essentiality prediction challenge. Breast cancer network inference, right. So how do you use proteomics or microarray time course data to infer networks. Again, network topology and parameter inference, whole cell parameter estimation, right. So this is something I was going to talk about. So the whole cell model of Mycoplasma genitalium. So this was the first organism for which a whole cell model was constructed by Marcus Covert's group at Stanford. It's a very interesting study wherein they studied several different processes happening in the cell and built one large integrated model for phenotypic predictions and so on. So maybe let's just go into one of these challenges. Yeah, this is how the whole cell model looks.
These are not incomplete challenges.
Huh?
These are not incomplete. Dream challenges means they want to do it or it has already been done.
No, so this is a competition that is announced from time to time. So these are very old. This is probably like 3 years old or something. So this is completely done. But it's a very good thing to benchmark what you do, right. So if you have a new algorithm which you think can perform better on this kind of data set, so it's a very good benchmark to go back and look at. And all these are attempted by the best groups in the world. So you have very good results to beat. And if you can beat those results, you really have something. So you will have you may even have access to the codes, I think, in some cases. You will have codes, what are the algorithms used, there's a very detailed paper. So each challenge culminates in one or more papers that describe how the challenge was solved, what were the different approaches, why were some approaches better than the others, right, and why was some approach not so good but still performed better than in certain aspects. Overall it may not have been the winner or whatever, but it excelled in certain aspects, right. So there are lots of things to be learnt when 100 people try to solve a problem, right. So it's really interesting. So, a recent problem that we actually grappled with was this really difficult problem of disease module identification. So, what you have is just a simple network, a string protein interaction network, go find modules, no other information is given. You do not even know what the proteins are. I just give you a network, A interacts with B, B interacts with C. Using the topology of the network, can you find modules that are enriched in disease? And it turned out that it was a very difficult problem, and from a very large network there are no team could predict more than 30-40 modules. Right? So, it was quite challenging, but then you are stretched to your limits, right. So, you try to pull all kinds of tricks to see if you can just predict disease modules using network topology. So, the inverse question is how much of information on disease modules is potentially embedded purely just in network topology. I think everybody understands and acknowledges that you need more biological information to build nice modules and so on, but what is the best you can pull out using this, right. So, that gives you a baseline of how much information is actually already present in that and so on. So, if you look at this, the number of processes are crazy. So, there is protein translocation, ribosome assembly, protein processing, translation, protein modification, complexation, transcription, activation, DNA repair, DNA damage, metabolism, so on and so forth. So, they actually modeled several different processes. They may have listed some of these here, and yeah. So, they had some nice awards and so on as well. Right. So, this was about nearly 5 years, 4.5 years old. Yeah. So, these were all the process. So, they had to initialize so many variables, send cell variables to all these processes or molecular functions and update. A really complex model, if not the most complex model that was built. This is something we understand very well, right. The metabolic model, that is nothing but an FBA based model, right. So, it was implemented using flux balance analysis, like huge number of parameters. So, but at least 30 different parameters were needed to be estimated. So, they have a scoring mechanism and so on and so forth. They have a leaderboard as well, which tells you how people are performing. So, which will get updated regularly. Yeah, look at the number of people who are involved in building this challenge, right. And these are the nice. So, if you see, there is finally this nice summary paper that was published as well. So, that will give you all the information that you ever wanted. So, let me throw some interesting questions. So, let us see if we can come up with ideas. How do you benchmark parameter estimation algorithms? He studied several parameter estimation algorithms, right. He studied simulated annealing, genetic algorithms, differential evolution, so on and so forth. Which of these would be the best parameter estimation algorithm?
That depends on the problem.
It often depends on the problem, but most of your biological problems aren't too different, right. So, you know that there are certain characteristics of your biological data, like they will be heteroscedastic. So, each point will have a different variance and so on, and you will have data of questionable quantity and quality, right. Under these scenarios, what would be the best model? What would be the best estimation algorithm? So, you need a very good, so the first challenge would be to have a very good cost function, but once you have a good cost function, you may have to evaluate what are the best. So, how does simulated annealing perform with respect to say genetic algorithms or so on, right. So, one way to do that would actually be you try and essentially simulate generate synthetic data, right, similar to what you would obtain in a biological scenario with known parameters, and then maybe add some noise and see if you are able to fetch back the original parameters, right. So, this is a very interesting way to figure out what would be the most practically useful parameter estimation algorithm. And of course, challenges like these are very useful to figure out what are good parameter estimation algorithms and so on. This is again a very nice paper which talks about simulating a living cell by Marcus Covert, who is at Stanford, who works a lot on whole cell modeling. Yeah, this is a nice history of whole cell modeling. Alright, so you had very simple models to begin with, Finally, there was E-cell, which could actually take 127 odd differential equations and simulate, but that was still in 1999. But beyond that, in 2012, we had the first whole cell model of Mycoplasma genitalium, and the model can accurately predict the activity of several enzymes. And could you make a, there are actually, you know, on the other extreme, there are virtual human models and so on, which are obviously a little more macroscopic, not as microscopic. This talks about single cell reactions and so on, and builds them towards a whole cell prediction. But if you're talking about the human, you have several layers in between, right? So the cells form tissues, tissues form organs, organs, and you know, interact with one another and so on. So there is a lot more complexity involved, but there are, you know, very good models of the heart, very good models of various systems and so on. Yeah. So look at this. So these are all the complex things that are included in the model. This DNA damage, repair, metabolism, RNA decay, protein processing, translation, so on and so forth, cell division. So all these processes are considered in an integrated fashion. So you should read. I have posted this paper, so I'm not going to go into too many more details on this. See, I think by definition, you are trying to manipulate the things that are not conserved in synthetic biology, right? So you probably wouldn't be tampering with, tinkering with central carbon metabolism and things like that. So you take any cell and you start working with it. It already has a conserved core, which you basically plug into, right? So you are saying you want to make a circuit where you want a core that is conserved or something. So I still don't understand the question entirely. What do you mean by you want to have a core that is conserved in a circuit? Why would you want it conserved?
Like a bow tie.
So because you want to make it very robust or something? What's the...
Yeah.
So there are actually multiple approaches to this. This is quite challenging. So one thing that you can't wish away is always evolution. You can't wish it away when you're building a synthetic cell. So there are strategies that people have been trying to explore to essentially slow down the rate of evolution rather than eliminate evolution, because you can't eliminate evolution, but are there genes that you can silence so that you can slow down the rate of evolution, right? So your system is going to be relatively more stable. But in the context of making a conserved core or something, I don't think that arises really, because you usually make an oscillator module or a sensor module and stick it into an existing system which already has a conserved core and so on. What you can do is you can insulate your circuit from unexpected behaviors. So I didn't go into details of this very interesting phenomenon called retroactivity, right? So what happens is when you have a system, you go and plug it into a context, just the fact that this is plugged into this causes some, even though there is no edge of this sort, there is an influence from the fact that this is plugged in here. But if you see, fortunately, something like the repressilator, even if you plug this into something, it actually retains its behavior, because the negative feedback loop is so strong here that it can still retain its behavior if it's plugged into a different context. So how does, right? So how do you account for these while designing a synthetic system? So these are very interesting open questions. So there are two ways to design a non-retroactive system in principle. First thing is you consider it during design and you build a system that is non-retroactive. Right? You know the context you are going to plug into and simulate it all along and figure out what is the best system that will, when plugged into this context, won't work. The other thing is you start with this network and you add another circuit to it, like an insulator. This is an additional device, literally an additional module that combines with the previous module and minimizes any crosstalk, any interferences and so on. Yeah, so what I was trying to say is that there is no question of engineering a conserved core or something, but I was trying to answer her, the reason behind her question. She wanted to design a robust system, a system that can reliably work, right? And one of the reasons we were talking about in a previous class was that it needs to have a conserved core, there is a particular bow tie architecture and so on. She was like, how do I build this kind of a conserved core? but what you do is you actually don't build a conserved core, but you want to probably build an oscillator or something like that, and then you combine it with an insulator or into a context by considering how the performance will be to make an accurate oscillator, a reliable oscillator. So in today's video, we covered a bit of ground in terms of the challenges in the field of systems biology. We looked at this very interesting paper called Bridging the Gaps in Systems Biology, which talked about the various challenges that are being addressed and still remain nevertheless. We also looked at the Dream Challenges website, which poses very interesting kinds of challenges for the community to tackle. So, these are very interesting problems for you to think of working on sometime in the future. We also looked at this emerging paradigm of wholesale modeling, and finally the concept of retroactivity, which is how do you reliably compose modules or circuits in a biological system, because you have a system that maybe oscillates and you stick it into a biological system, it may no longer oscillate, right. So, how do we build reliable models, and can we account for that in the modeling stage itself, so that when you do your synthetic biology it works quite well. So, in the next video we will recap the entire course, beginning right from how you model the fundamentals of modeling all the way up to the concepts of say metabolic networks, regulation and synthetic biology.